this webinar here today. I'm very pleased to be able to introduce to you today Steve Gygi, who received our annual Don Hunt Award. This is a distinguished contribution in proteomics. And Steve definitely is an amazing awardee for this. So I would like to say a few words about Steve and his very impressive career. So Steve Gygi received his PhD from the University of Utah in pharmacology and toxicology, performing small molecule mass spectrometry. He went on to pursue his postdoctoral work then with Rudy Ebersold at the University of Washington in 1996, working in a new field at the time called proteomics. In 2000, Dr. Gygi moved to Harvard Medical School and joined the Department of Cell Biology. Currently, he is the faculty director of two MS core facilities, which is quite impressive. The Taplin Biological MS Facility and the Thermo Fisher Center for Multiplex Proteomics. Research in Steve's lab centers around developing and applying new technologies in the field of mass spectrometry-based proteomics. These include the systematic and proteome-wide measurements of many protein properties, including their expression levels, modification states, structure localization, function, and interactions. So quite comprehensive. For example, the Gygi lab, together with the Harper lab, is creating a genome scale map of the protein-protein interaction landscapes in cells, termed bioplex. In addition, sample multiplexing techniques like tandem mass tags, TMT, are being improved to allow Steve and his lab to 16 proteomic samples to be analyzed simultaneously using high-resolution mass spectrometry. Well, with this, Steve, I would like to welcome you today, and I'm really happy that you can start off our webinar with your talk. So please welcome Steve, everybody.
Thank you, Brigitte. Yeah, before we start, I just want to acknowledge that we're not meeting under ideal circumstances here. We're clearly not in Seattle, and most of us are distanced from our labs, and life is just really not normal. I just want to say I wish you the best to you and your loved ones in this most trying time, and hopefully we can all stay safe and stay at home. I know that we'll get through this, and soon we'll be back to the work we like to do, which is unraveling the mysteries of the proteome at with an even greater appreciation, I guess, for our shared community. So I'm very much looking forward to sharing some time with you and highlighting the role that my lab has played in the success of these isobar tagging strategies. But if you look at this slide, you can see that I guess not everyone likes TMT. This is Aquaman here. Apparently, I never knew that Aquaman was a label-free guy, but I guess he doesn't really like TMT. But actually, TMT in this case stands for the 30-meter telescope, because as we know, I think almost everyone loves TMT, as this picture shows here. So it's my great honor to be awarded the Don Hunt Distinguished Contribution in Proteomics Award from US HUPO. Throughout my career, I've always looked up to Don Hunt for his approach to science. He loves instrumentation, as you can see him here in this picture. He loves teaching, and there's just so many things that kind of go back to him, including FTICR, really commercialization of FTICR came through him, the discovery of ETD. In application, he's a self-trained immunologist with his work with MHC peptides. It's just really important. It has a lot of biological meaning as well. And of course, the entire field of epigenetics and chromatin biology is based on some of these ideas of the histone code that came from the Hunt lab. But maybe the most recognized thing from his lab, I guess, are some of the people that came from his lab, which continue to kind of prosecute proteomics in the Hunt way. And here's just a few examples of people from his lab. I just really hope that in my career that I can so successfully marry, I guess, chemistry, biology, and instrumentation as Don has done during his. So if you know me, you know that my favorite person in science is Sidney Brenner, and my favorite quote from Sidney Brenner is this quote here. And it says that progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. So we recognize Sid Brenner as kind of the father of molecular biology. He proposed the concept of messenger RNA. He discovered the triplet nature of the genetic code. And really the central dogma of biology goes through him. So, you know, he wasn't awarded the Nobel Prize for any of these really amazing discoveries, but he had to go back and reinvent himself. And in 2002, he got the Nobel Prize for his model organism, which was C. elegans, this little 1 millimeter worm that is transparent and allowed him to call it nature's gift to science. But I like that he thought that progress in science really depends on new ideas, which lead to new discoveries, new techniques, which lead to new discoveries, which lead to new ideas. So if we want to talk about multiplexing today and sample multiplexing in general, the origins of sample multiplexing in proteomics go back pretty far. I guess, you know, there was a lot of MS1 based multiplexing that kind of happened. And here's a paper from the turn of the century. I happen to be an author on this paper. It's from Rudy Aebersold's lab. And we were chemically labeling two different samples. And these two have differences in stable isotopes that would be that would be derivatized to them. And then after they were mixed together here, this is where the multiplexing happens. In the mass spectrometer, each peptide would have two different peaks, and the abundance of those peaks would then represent the amount in the original mixture. And this was more formalized by metabolic labeling by techniques like SILAC, which allowed for multiplexing and quantitation of two samples again, and maybe even three. But what we learned from MS1 based multiplexing like this was that because it requires duplication of each peptide, that when you combine more than a couple, you really have a problem with the complexity, and the depth of coverage is just really affected by this. So if you really want to look at true kind of the origins of large scale sample multiplexing, you have to go back to this paper here by Andrew Thompson. Here's a picture of Andrew Thompson here. This paper is the first report of an MS2 based strategy, which they called isobaric labeling for quantitative proteomics. And there was just a duplex is all that they demonstrated in this, and it's a peptide based reagent, which was called the tandem mass tag. Right behind this paper, what came out a paper which demonstrated something called iTrack reagents, which had a 4-plex quantitation and also used this MS2 based approach. So even though we've had new reagents and new instrumentation and new methods, these two papers really represent the foundation for pretty much all modern proteomic sample multiplexing. So how does it work? So if you think about it in a 6-plex format here, you might have 6 samples. Those samples might represent 6 livers from a mouse, or 6 cell lines, or 6 plasma samples. But each of these 6 samples then is labeled with one of the 6 TMT chemical reagents. After labeling, they're mixed together, and in the mass spectrometer, they don't have any difference in the mass because they each are offset by the same mass by these reagents, and thus they stack up in the MS1 scan. So already there's a difference between what we saw before in the MS1 based sample multiplexing, in that there's no more complexity added to the sample. And in fact, if you could get to 96 samples, if you had the chemistry to do that, they would stack up and it wouldn't be any more complicated than a single sample. Really attracted me to this type of sample multiplexing in the first place. And there is a difference that can be revealed here under fragmentation conditions in an MS2 spectrum, for example. One can then reveal differences based on stable isotopes that allow one to quantify from an MS2 scan the abundance of what was present in the original mixture. So here's the structure of the TMT reagents. They consist of a reporter ion, which is the red part here, it's cleaved. There's a green part here, which is the balancer, which has balances out the stable isotopes. And then there's a reactive group here, which will give you an NHS ester, which will allow for derivatization to either peptide N-termini or to lysine, the epsilon amino group of a lysine. So the real business of the TMT reagents are these asterisks here. So if you look at these asterisks, there's 5 of them. And those 5 asterisks are 5 positions for heavy atoms, for example, carbon-13 or nitrogen-15. And you can see the 126 reagent shown here. If you cleave here at this bond here called the HCD cleavage bond, then you release the reporter ion, which has none of these carbon-13 heavy atoms in it. And so it has a mass of 126. If you look at the 127 reagent, by cleaving here at this bond, one of those asterisks is then placed on the other side, and it ends up allowing for the 127 reagent to have 1 Dalton more in mass. All the way down to the 131 reagent here, which has all 5 asterisks here are on the other side of the bond, and so the reporter ion then has an additional mass of 5. And so this is how the original 6 TMT reagents came about. But if you really want to understand TMT, you need to meet these two people. So this is John Rogers and Ryan Baumgarten, and they are at Thermo Fisher in the Rockford, Illinois site. They are originally from the Pierce Chemical Company. So John Rogers is, well, I guess Ryan is in charge of developing the methods and making sure that they work for TMT. And John is really responsible for shepherding TMT throughout the commercialization process. So if you like or don't like TMT, it's John's fault. He's the one who has been in charge of disseminating TMT. He hasn't got distracted. He's been very good at making sure. He flirted a little bit with new code for a while, but I think he loves TMT as much as I do. So maybe I'll go through just a couple of the papers that I think were most important in really making TMT available to everyone. And the first was a paper by Willie Haas and Lily Ting here back in 2011. And what they had found was that, well, the TMT reagents are reproducible in the measurements that they make, that they are really not as accurate as you would hope they would be. There was something called ratio distortion that happens where if you expected to see a 10-fold difference, you might only see a 4-fold difference. And so Lily and Willie set out to kind of solve this problem, ended up in this paper that you see here. And the idea that they proposed was that through an additional step of gas phase fractionation, that one could take not reveal the isobaric labeling reporter ions in the MS2 scan, but to reveal them in the MS3 scan by isolating a new precursor, gas phase separation, interference-free measurements down here. And they found that this really worked. But almost as soon as they had kind of restored the accuracy, we noticed that when you isolate a single precursor and collect an MS3 scan, you don't have very much signal that's left. And so we felt like the sensitivity was really going to be a problem and that the MS3 was going to have issues. And pretty much at that point, Graham McAllister showed up in the lab pretty much the next day. And Graham McAllister is the academic grandson of Don Hunt. And he came from the Kuhn lab. So he knows instrumentation. So, you know, Graham's idea was to say, look, if you want to get more sensitivity, you don't have to isolate a single precursor. You could isolate multiple precursors from that MS2 scan using something called a notched isolation waveform. So this multi-notch waveform could be applied, and that would allow us to isolate multiple precursors at the same time. This had no cost whatsoever to the instrument time, and that this was then termed synchronous precursor selection and became commercialized in an instrument. So this really did make a big deal because it ended up being about an 8-fold change in the average amount of signal that we were seeing in our TMT reagents. And so I can tell you today, we wouldn't be doing MS3-based TMT if we did not have the notched waveforms available in ion traps. So Graham wasn't finished. We also noticed that when one of the reagents was resynthesized, this is the 127 reagent, you can see where this asterisk is here. It's just on this bond and it gives you the 127 reagent. At one point it was resynthesized and it was put on the nitrogen as an N15. And when we looked at these in the mass spectrometer, they weren't the same anymore. They had a different mass. So they were off by about 6 millimass units. So this may seem like a very small number. It's the third decimal place and it's a 6 at that position. But this difference is fully resolvable in an Orbitrap mass spectrometer. And so with that knowledge, Graham proposed that one could resynthesize these 4 reagents using N15 at these positions here, and that would give you 4 new reagents, and you could jump from 6 to 10-plex. And pretty much overnight, there could be a 10-plex version of TMT. And then over a couple of years, we convinced John Rogers again at Thermo Fisher that it would be time to make an 11th reagent. And so this is how the TMT11 came about. So now we have a platform that we use, which has a number of samples, up to 11 in this case, or 16, as I'll show you in a minute, that through a process, one can extract the protein, turn that into peptides, label those peptides with these TMT reagents, and then mix them together. You have some kind of an unholy mixture here of proteomes, but that mixture then is so complex that it's difficult to analyze that all at once. And so we fractionate that using basic pH fractionation and shoot 12 fractions. So at the end of the day, we have 12 autosampler vials. Those 12 fractions end up using 3-hour gradients on the mass spectrometer, and this takes about 36 hours. So we use 25 micrograms of starting material, we use 3-hour runs, and 36 hours later, we have about 8,000 proteins, which are quantified across all 11 or all 16 of the samples. So why do we use sample multiplexing? And I think there's lots of reasons why you might think of this reason here as the #1 reason, and that is that you can increase the throughput by using this. If you had 500 patient samples, divide that by 16, it's a whole lot less than 500. But we also really look to multiplexing as an advantage because it has no missing values. Everything that's contained within a single plex gets quantified against everything else. And this is really a wonderful thing when you see it happen. Finally, because it's based on stable isotope dilution theory, the quantitation really provides kind of unmatched precision. It's just remarkable to see how good the quantitation can be. But I guess the main reason that I like TMT and that I'm still a big fan is that it really facilitates complex experimental designs. And by that, what I mean is that you can include within the single plexing experiment, you might include biological replicates of each condition, dose response, time series data, rescue experiments, positive controls, negative controls. You can include a lot of different things in that experiment. All of those are quantified at the same time. And it really helps to give you a lot more biological meaning. And so the way we kind of talk about it is that proteomics experiments turn into the world's best biological assay when you do this. It's not just that 11 to 16 gives you a 40% increase in channels, it's that 11-16 provides you with an even better biological assay that allows you to see the entire proteome across these 16 conditions. So maybe I'll highlight one more feature here that has happened in the past, more recently, and that is that some work by Chris Rose and Brian Erickson here, where we kind of knew that MS3 scans are fairly time consuming, and that on average less than 50% of all the MS spectra that we collect are confidently matched. And yet with the SPSS method, every MS2 scan ends up having an MS3 scan attached to it. So when you look at the final data set, only about 1/3 of all the collected MS3 scans are actually used in the final data set. So this seems very useful, very wasteful, because there's so much time used for the MS3 scans. So the solution that we've been thinking about is that if we could know the identity of the peptide in real time on a millisecond level, then we could only collect MS3 scans when we knew that they were going to be meaningful and going to end up in the final data set. And a real-time database search might allow us to do that. So Chris Rose went ahead and collected, and he basically rewrote sequest, Jimmy Yang sequest in Lua, and kind of demonstrated to us that this would really work. And then Brian Erickson came along and he used an API which allowed us to listen to the data stream from a Fusion and to insert MS3 scans. And Brian Erickson published in this paper, nice paper here, a demonstration of using that API. So we then turned to Jimmy Yang, who was at the University of Washington. And Jimmy Yang had originally written sequest, and he had also written an open source version, which is called Comet. And we challenged Jimmy Yang. We said, look, we don't want a data set level comet here. What we want is a single spectrum comet that's as fast as it can be. And a couple of weeks later, Jimmy came back and said, okay, here's my version of a single spectrum comet. And we knew immediately when we saw these data that it was going to work, because it's a human database here, the full human database. It's forward and reverse sequences plus contaminants, 3 different isotope windows because we didn't know the monoisotopic peak that well. And it was still only taking about 7 milliseconds on average to do these searches. And yet, when we added modifications, it was only at, going to about 17 milliseconds. So we knew if it was less than 50 milliseconds, we knew that this was going to be a very successful strategy. So on the external computer, we can now do these searches and then insert back into the mass spectrometer the MS3 scans when we want to collect them. So to reformalize this a little bit better, we plugged in, again, a piece of software that we call Orbiter into the Orbitrap mass spectrometer here through this API. And it contained several things, not just the Comet algorithm that Jimmy had given us, but also some pre-processing work to look at the monoisotopic peak, as well as look at the false discovery rate here and try to get the best answer that we could get. And this was all done by a postdoc in the lab. Here he is as a graduate student. His name's Devin Schweppe. And then as a postdoc in my lab, he looked like this. And now that he's at the University of Washington, he's got a new picture and a new look, it looks like. And I think it looks more like the cover of a smooth jazz album than it actually looks like a picture, you know, for a faculty member. But hey, everybody has their own ideas. So what Devin's really noticed is that using the real-time search has these two really, you know, big advantages. The first is that you reduce the analysis time by about half of the same at the same proteome depth. So what this means is that instead of 36 hours to collect the 8,000 proteins across the TMT experiment, I can do it in 18 hours. My 3-hour gradients become 90-minute gradients, and I get the same data. And also that because I know the peptide sequence, I can make sure that my SPS ions that I'm going to target for the MS3 are really the B and Y ions from that peptide. And so I get even better quantitation out of this. Here's an example of it running. And what you can see is it's about 4 seconds, and there's about 70 MS spectra that are going to come through here. This looks like a tune window to you, but it's really just listening to the data stream and plotting it out in the orbiter. And over here, you're seeing the peptide that's being matched as you go along. And every time there's a color that's shown on here, it's going to be a peptide that's identified and matched, and it's going to throw an MS3 in here, and the MS3 show up as just kind of sticks in here so fast in the bottom part over here. So this is how orbiter looks when it's running. And here's the final results from looking at an experiment with orbiter. If I take these 3 cell lines, HCT, MCF7, and HEK cell lines in replicates here in this 10-plex, if I use 12 fractions to analyze the TMT labeled versions of these, what I can see is that it takes around 36 hours. I can use 18 hours, and I get about the same number of proteins that are quantified, whether I use 36 hours or whether I use 18 hours. And so this is kind of the way that we really look at how to use this better, is that we really get about twice the instrument efficiency. And so when we use our Eclipse instruments now, we really notice that by only using half the gradient length, they're open a lot more. And Devin has published this now in a paper in JPR. So let me show you why we get a little bit better quantitation as well. So on the y-axis here, the SPS-MS3 ions, SPS-MS3 data for each protein, showing you the MCF7 versus the HCT116. And then on the x-axis here are the orbiter results for those same proteins. And what you can see is there's a very good correlation here, but there are some that are kind of a little bit off the axis here and off axis here. And each of these are where we have a little bit better quantitation because of the real-time search. I'll just highlight 2 extreme examples here, where endoglin and ALPK2 are kind of known MCF7 kind of specific proteins. So you can see in the real-time search that there's way more of these showing up in the MCF7 cell line compared to the other 2 cell lines. But when we looked at it with the SPS-MS3 for these 2 proteins, it didn't do quite as well on endoglin, and it really didn't do well at all on the LPK2. It's likely that there was a fragment that was selected that wasn't from an actual B or Y, and that gave us kind of a poor quantitation event. So you just get better quantitation event when you use a real-time search as well. So let me go on and talk about the 16-plex now with the last 10 minutes that I have here in the talk. And so, you know, when you're looking to understand why 16 is the right number, I think you can look at this here. This is where a person named Edward Muybridge was interested in creating, it was 1887, they're interested in moving from pictures to, say, moving pictures. And so they were trying to figure out what was the right number of pictures to help you look at something that's very dynamic in a moving situation. And so he settled on looking at a racehorse, and what you can see here is the result of that, where he came up with the idea that there should be about 16 pictures would be the right number. And it's actually just beautiful. I really like to look at all 16 of these and to see the differences. And when you put these together, it gives you the full view of the entire system of this horse. So if we think about the TMT, 16 is the right number, what does the competition for TMT look like? Well, label-free would look something like this, because, you know, it's a single shot and a single picture. If you wanted to shoot multiple label-free samples, it might look like this. This just shows you that there's some missing values that show up in there and makes it difficult to interpret. SILAC would look like this. iTrack would look like this, kind of a choppy horse. A 10-plex is looking a little bit better. And a 16-plex, very smooth. It looks very good. So we like the 16-plex. So there's these new TMT 16-plex TMT Pro reagents. They're a little bit larger than TMT, as you can see here. And the reporter ion group now is based on this pyrrole structure here with an isobutyl group here. So you can see that this looks like a proline, this pyrrole group here. So they're named pro because of the proline group that you see there. So they're about 25% larger in the normalization group here. So Proteome Sciences made these reagents, and Thermo Fisher is distributing these reagents. So when we got the reagents, there was 16 tubes that we received. They look like this. They probably look a little bit better when you get them from there. There's also a 0 reagent that you could use for method development. So 16 tubes and 16 reagents. So I asked Jean-Man Lee, who's a postdoc in the lab, and Joao Paulo, who was an instructor in the lab, to kind of work these up and to look at this. And the paper that they came up with is shown here, just came out a couple of weeks ago in Nature Methods. And at the end of the day, you can read what I've highlighted here, that they ended up showing that they could quantify 8,800 proteins across an analysis time of only about 67 minutes per proteome using the 16-plex. So they start with 25 micrograms, they quantify more than 8,000 proteins, and they barely use an hour, per proteome. So it's a pretty remarkable advance to put these all together. So here's a little bit more about these reagents. There's 9 positions for heavy atoms here. So you can see all 9 positions here. And these are the first 6 reagents here that I'm showing. So you can see that with 9, we actually could make up to 18 reagents. They're showing you 16 here. So it could be 2 additional reagents coming forward. And if they wanted to redo this reporter ion structure and add a couple more carbons, you could get 21 reagents out of this structure. So there is potential to grow. So maybe one thing to understand about these reagents that'll really kind of help to kind of offset them is to show you this part just right here. And that is with the TMT Pro reagents, they fragment at lower energy. They fragment better than the original TMT reagents. And so at 45% energy versus 55 for the maximum that you would get here. But also there's an increase in signal. You can see there's about a 40% increase in signal across here. This was just key for us because if you take a microgram that you can load on the column and you divide that into 11 samples, or you divide that into 16 samples, you're losing about 40% of your potential signal when you multiplex up to 16. But because there's an increase in the signal because of these reagents, it's pretty much a wash. We really don't see a difference in the signal, in the sensitivity between using the TMT0 and using the TMT Pro. And pretty much everything else is the same. You need to adjust your labeling amount by about 25% more. The MS3 times are actually shorter. It's a little bit more signal, so they're a little bit shorter. Your retention time is slightly longer, maybe up to a minute or 2 longer for the retention time because they're a little bit larger. But everything else will feel very, very similar. And we usually label about 25 micrograms for all 16 samples. Let me just show you one example here with the last, looks like I have 5 minutes left. I'll show you this one example. So in proteomics, we're interested in discovery, in the drug discovery part of proteomics. We're interested in target identification. And so many times we have a compound, a natural product, or a drug, and we're interested in understanding what is in the proteome. And so one way to figure that out is to use something called a thermal shift assay. And what that means is that if I take a native protein and I add temperature, at some point it will begin to denature. And that's these kind of purple, ugly things here in the middle of the protein. When it's 50% denatured, and then all the way down to where it's 100% denatured and aggregated. And so this melting temperature curve that you see here is unique for every protein. But what might happen is when you add your drug, you might impart more energy and you might change the temperature at which it will melt. And so these thermal shift assays, they're often something similar to this, where dihydrofolate reductase here is the target of methotrexate. And you can kind of see here that this melting curve for DMSO versus the melting curve for methotrexate against this particular protein shows that it's quite a bit longer. You can quantify that in something like is shown here from this paper down here. So one of the ideas there was to say, can we formalize this a little bit more in a 16-plex, and what can we learn about it? And as we were doing this, we started noticing how much work it really was to do this, because you have to use 20 channels. You have to use 2 10-plexes, one for the untreated for the 10 temperatures, one for the treated for those 10 temperatures. That's 20 different channels. You put those 20 channels back together into 2 separate 10-plexes and analyze those, then you draw the curves. There's a lot of work that's involved in that. Well, about this time, we saw a paper from the Zubrov lab that came out And it had a new assay that they call PISA. And in this PISA assay, PISA stands for Proteome Integral Stability Alteration Assay. The idea here is this word here, integral. The idea was that you don't need to create the curve. You don't need to measure every single point on here to make the curve. What you really care about is the shift. And by taking the area under curve, if I just combine all the samples into one and then measure the amount of protein, I can look at the shift that's there between the treated and the untreated, and that becomes a single measurement and only two channels, one to measure the untreated, one to measure the treated. Thus, I can use the remaining channels to measure the area under the curve for replicates. If you use a lot of replicates, you can create a t-test basically. So you went from drawing curves and using fancy software for the curves to just doing a t-test to look for potential differences that are in here. And so we wanted to John wanted to take the Zugroff version of PISA and to create it into a 16-plex. And also he wanted to shorten, for other reasons, he wanted to shorten the region of the temperatures that we were using so we could get maximal shifts in the area under the curve. So this is a busy slide, but it's kind of what John did. I just want you to focus over here, where at the end of the day, we have 16 samples. Each one of these is going to have 10 different temperatures that are going to be in there, and they're going to act like replicates. So I have 4 replicates of a vehicle. I have one compound that I have 4 replicates of. I have another compound that I have 4 replicates of, and I have a final compound that I have 4 replicates of. All of that ends up in the same experiment. And so you went from needing 2 10-plexes to do a single compound to having 4 replicates of each compound and 3 compounds. So it's orders of magnitude better in what this particular assay can do. And what you're looking for now is a t-test between the gray here and the green, where you're asking, are these significantly different from each other, the melting temperature significantly different when I apply the drug to the lysate. And here's just an example of one of these. We'll zoom in on these 8 channels here. If you use NADH as your compound and you're interested in what binds to NADH, you will see big shifts. Like this is a big shift in the melting temperature, in the area under the curve for the melting temperatures. And the most abundant protein here is the actual protein that synthesizes NADH, and it uses ATP and nicotinamide to make NADH. And there's many others, hundreds and hundreds of other proteins that bind to NADH, and we see many proteins that are shifting based on this assay. So the final thing that we did with this was we wanted to apply it to something that had lots of targets. So we looked at either wild type lysates where we had sterosporin had been applied, or where 20 micromolar sterosporin had been applied. So 2 different doses of sterosporin with 5 replicates of each. And what you can see here is that the shifts are not very big. They're pretty small, but there are, many dozens and dozens of proteins that are all kinases which are shifted. And that's just showing that it's working in either the 1 micromolar or the 20 micromolar stage. And it's just another example. You can also look over here at just the 20 micromolar version of this, and you can see that there's about 80 kinases which have a p-value which is adjusted and is high enough to say that these are targets of sterosporin. So, again, I think this is going a long way to showing how the 16-plex can be put to use to look at many compounds at the same time under many conditions. And I just really like that example. So I've shown you that a real-time search you can use this SPS-MS3 workflow, and you can reduce your analysis time by 2x. I've talked to you about these new 16-plex reagents, how they're powerful tools. I've showed you that there's no missing values within the same plex. Proteomics, within a plex then represents what I think is the world's best biological assay, because everything is compared against everything. And of course, nowadays, if you wonder what we're working on targeted TMT proteomics workflows. For example, Devin is working on FAMES in targeted fashion. Ching has a new paper that's coming out next week where he's taken this Tomahawk targeted TMT assay, and he has used the API to make it even better. So this is where we're kind of headed in the lab now. So I'd just like to thank everyone that helped make this possible, including people from my lab, Thermo Fisher, John Rogers and Ryan Baumgarten. Again, here, people at Proteome Sciences, Andrew Thompson, Ian Pike, and Carson Kuhn for their work, as well as the funding from Google Ventures, Third Rock, and NIGMS for the PISA work. And of course, 2 people in the lab that have, always been part of everything keeping me straight. Ed Hutland, who is a bioinformatics guy behind most of the things that happen in the lab, and Joao Paulo, who's in charge of instrumentation in the lab, and Edward Muybridge for helping me learn that 16 is the right number. So with that, I'll stop and look forward to doing some questions.
All right. Well, Steve, thank you so much. This is really wonderful. I think it really shows how you use technology and latest newest developments to apply them to some really interesting biological questions. So thank you for that. We have a couple of questions here, but maybe I could start up with one question. I was actually wondering, over the years that you've been doing mass spectrometry, what do you think is your what do you think is the bottleneck, and where does it change? Is it sample prep, instrumentation, processing? Maybe you could comment on that.
I mean, I think always the real bottleneck that everybody thinks about as we move for TMT, I don't know about proteomics in general, but the real bottleneck for TMT is likely to be sample prep, because the channels do affect each other. And so if you have better hands or you have a better method, then you're going to get better results out of the TMT experiments. And so, you know, we and others are putting on, this guy right here, Ryan Baumgartner, you know, are putting a lot of effort into making sure that the sample multiplexing is good, because as you combine more samples together, it has to get, you have to have, you know, really excellent multiplexing for the sample handling as well. So I think that's the major bottleneck.
Okay, thank you. So we have a question here from Christina Chiva. She asked about TMT Pro reagent stability.
Yeah, so we looked at the TMT reagents stability. They, you know, Thermo Fisher had their, what they always do, where they, you know, they look at how long it lasts in the freezer, and they take it to have 70% of the original activity, you know, months later or something like that. And so we find it to be, if they're asking about stability like in the freezer, or if they're asking about stability to fragmentation, you know, we have not really noticed a difference either in the X scores or in the fragmentation patterns. It is a larger reagent, but it really hasn't been something that we've noticed. So if she's talking about stability from a reagent standpoint or stability in an MSMS standpoint, I think both of those are not really things that we have had issues with.
Okay, good. We have a couple of questions, both from Rex as well as from Ying Zhu, and they ask about TMT data normalizations, tools for those, and how you also normalize across multiple TMT data sets.
That's really a great question. So normalization is probably one of the key things in TMT experiments that you need to do. So we always do what we call a column normalization or a protein normalization. So we take the total signal that we see across all 16 channels, we add that up, and then we normalize back. So that will usually be different by 5% or 10%, and that represents pipetting differences. So they become really accurate because you're able to normalize back away from kind of pipetting errors or sample handling differences there. So that's the first normalization step that we do. And if you want to get across plexes, there's two ways to do that. The first is if you have replicates, you can put the replicates in different TMT experiments. So if I have 16 cell lines and I have 3 replicates of those 16 cell lines, I would do 3 16 plexes, but I would have each replicate in a different plex. And in that way, I would have all the measurements I needed to make in each one, and I wouldn't need to get across. I would just do the measurements within. Now, if you do need to get across, then of course we recommend using a bridge channel, where if you had 500 patient samples, you might mix together a fraction of those or all of those and have one channel, the 16th channel, that you might add back to each one of those. So I think that's the way we normalize based on, you know, protein amount in order to get it there. And then we would use a bridge channel, or we would use the replicates to separate the plexes.
Okay, thank you. Yeah, so there's another question here from James Sanford. He asks, he's wondering if you could talk about missing data across TMT plexes and how to navigate that issue. Basically, within a plex, missing data may be low, but between 2 plexes, overlap could be poor. He mentioned specifically phosphoproteomics.
It's absolutely true. When you move between different plexes, you run into some of the same issues that you run in with others, and that there can be missing values. So don't think that because I say there's no missing values, that the problem is solved. If you have more than one plex, you run into, you know, an issue where some things might be missing from that. Now, this can be especially a problem when you're dealing with data sets where you care about the peptide level, for example, phosphoproteomics, as mentioned. And so, there's really no better way to handle these other than to say you can move to targeted methods, or you can, scale up and do fractionation. You can do whatever you want, but at some level, you're going to have to deal with the fact that there are going to be missing values between them. Some people are trying to come up with new ways to impute, to impute the values that for those missing values. But it's just something to be aware of, that as you go across. Now, it's not nearly as big of a problem at the protein level, because we get 8,000 or 9,000 proteins that are quantified, and most of those show up. The vast majority of those show up in all the way across. So it's not nearly as big of a problem at the protein level, but as mentioned by the question, at a peptide level where you care about phosphoproteomics, it's more difficult. And so you might need to move to targeted strategies where you could look at just look at the phosphorylation sites that you care about, and you won't miss those then.
Mm-hmm. Thank you, Steve. We have another question from Antrix Jane. The question would be, does increasing the number of channels have pronounced effects on multiple channel carryover, affecting sample quantification?
It doesn't really. So there's, they're pretty small. They're 126. So there's 6 or 7% carryover for the carbon-13 into the next channel. But most of that is just taken care of by, by correcting for the isotope and the impurities in the synthesis. So, we don't really see that as an issue. I mean, some of the ways to check that, of course, are to leave those channels blank and to see if the software then puts significant signal into those channels for proteins. But in our case, it hasn't been any different than the 10-plex, and we haven't had an issue with having additional channels, because again, they really contribute to the one right next to it, just like the 10-plex did.
Okay, thank you. And then, we have a couple of questions. They are asking, have you looked at using less than 25 microgram input material? What do you think might be the lower limit, or what do you feel comfortable in your lab to use?
Well, again, if we like to fractionate, and so at the end of the day, you're going to be putting whatever 16 samples you combine onto some type of fractionation. So we have labeled 10 micrograms. We have labeled less than 10 micrograms. We stick with 25 micrograms because it just makes everything so much easier, and we have multiple shots as we go down through there. But it's a good question, but we're certainly comfortable at 10 micrograms as well as at 25 micrograms with the labeling.
Thank you, Steve. Thank you for answering really all these questions. We can see that there's a high amount of interest in your research and in the tools that you push forward and publish about. There are actually a couple of additional questions in the question and answer sessions, and maybe Steve could, during the day, start answering some of those. And I do think this has been a really wonderful conversation with you, Steve. I really would love to really congratulate again, and thank you for taking your time today. And this has been a great start. And we would like to now continue with some of our sponsor research. And I will move over to David Arnott from Genentech, who will introduce our first sponsors. David.
Hi, good morning, everyone, or afternoon, or evening, as your time zone may be. It's my pleasure now to introduce the first of our sponsors, that is Thermo. They will be giving a 30-minute webinar followed by questions and answers. Many of you will know Thermo as the manufacturer of high-performance mass spectrometers, but over the last decade, they've put together a full ecosystem of instrumentation, equipment, reagents, and sample preparation kits for supporting life science research. They're a longtime sponsor of US HUPO, one of our major sponsors, and we are very grateful for all the help they've given us over the years.